There are different important sites in DNA which are involved in the transcription control. And the transcription control mechanism is a very, very complex process. In this segment, we will study the different sites which are involved in transcription and how they control the transcription by their mechanism. Now, as said earlier, the complexity of the control of gene transcription that can be illustrated by examining the DNA in and around a single gene. A gene and the DNA which is in that gene, the DNAs which are around that gene, and how they take part or play their role in controlling the transcription this. Now, the important example, for example, we can study, that is the phosphoenol pyruvate carboxylase carboxykinase, PEPCK. And the gene that actually controls or encodes for the phosphoenol pyruvate carboxylase, that is the PEPCK, is a good example to study the role of certain DNA sequences in maintaining the expression of this enzyme. That's how this enzyme is expressed at certain level, and that is the example which we will study. Now, this enzyme is synthesized in a liver where glucose levels are low. Conversely, synthesis of the enzyme drops sharply after ingestion of a carbohydrate-rich diet such as pasta or sugar, sweets, or other things. So this enzyme level, they drop down very considerably. Now, the level of synthesis of PEPCK messenger RNA is controlled by a variety of different transcription factors because, the level of synthesis of PEP messenger RNA, that will be controlled by different transcription factors, and they include number of receptors for hormones that are involved in regulating carbohydrate mechanism. Because in carbohydrate metabolism, they are controlled by the hormones, and the different transcription receptors, they control those hormones and the enzyme factor. The key to understanding the regulation of PEPCK gene expression lie in, one is unraveling the function of the numerous DNA regulatory sequences that reside upstream from the gene itself. That are those receptors which are actually residing upstream of the gene, on the other side of the gene itself, and they control the production of this. Then identifying the transcription factor that bind these sequences. Those transcription factor which are binding these sequences, to identify them. And third and last is elucidating the signaling pathways that activate the machinery responsible for selective gene expression, जो कि इस जीन को express करते हैं. Now, the closest or the most proximal regulatory sequence upstream of the PEPCK gene is a Tata box, which is the major element in a gene's promoter. Gene का जो promoter है, उधर Tata box is there upstream, and that controls the PEPCK synthesis. Now, the sites at which transcription factor bind to the region flanking the PEPCK genes, they are called response elements. So the response elements are responsible for controlling the PEP-CK gene, and these are the different factors which we have studied earlier in this segment, which control the which are involved in the transcription control of PEP-CK messenger RNA.